Hello, everyone, and welcome. Welcome to this audio podcast today. This audio podcast is part of the Serology Squil. This audio podcast is part of the Hustle and Serotype. This audio podcast is part of a Defcon premiere, and this audio podcast is part of a Storm Robotics production. Following on, and continuing in around aspects of the intro of this audio podcast premiere, looking at chemokine receptors and leukocyte trafficking in human acute allograft rejection. So so secondly, chemokine receptors differ in the importance as targets in allo responses. Thus, chemokine receptors such as CCR1 and CCR2 are primarily promote macrophage recruitment to an allograft, such that targeting in laboratory knockout mice or murine models have only a modest effect on allograft survival. Thirdly, the effects of containment immunosuppression can modulate some of the rather sort of grayish sort of outcomes of chemokine receptor targeting in untreated laboratory mice. Thus, the use of subtherapeutic regimen of immunosuppression, that of calcineurin inhibitors or CNIs, such as sort of the older immunosuppression, such as cyclosporin A, has a synergistic advantage with targeting of chemokine receptors, which by itself has only a weak or negligible for example CCR1, or negligible for example CX3 CR1 advantage. In the case of chemokine receptors, with the most impressive experimental experimental data to its role in allograft rejection, expression of CXCR3 and its ligands is closely associated with with AAR clinically. Data had emerged. Data had emerged, for example, from 163 renal allografted recipients concerning the extent to which chemokine and chemokine receptor polymorphisms can affect the incidence of AAR. To add here, to also add or to iterate in as part of this audio podcast, and in as part of this audio podcast, some of the sort of the some of the main some of the main sort of functions, if you like, functions or aspects in around chemokine action include for example, where chemokines are released by stimulated cells that act locally. Chemokines are produced by intrinsic cells as well as by infiltrating cells. Chemokines act through G protein coupled 7 transmembrane receptors and induce calcium influx. Chemokines also bind to proteoglycans and extracellular matrix. Chemokines induce hepatotaxis cell migration along surface gradients. Chemokines also can activate adhesion molecules and can induce oxygen burst and the release of granules and proteinases by leukocytes. And chemokines control aspects of immunomodulation, angiogenesis, hematopoiesis, and development. So that draws this audio podcast to a close. This audio podcast is part of the Serology and Squil. This audio podcast is part of a hustle and Serotype. This audio podcast as part of a Defcon premiere, and this audio podcast as part of a Storm Logistics production.